In 2011 and 2013, two large clinical trials, AMHI and HPS2, Thrive, respectively, delivered what appeared to be devastating verdicts on niacin's cardiovascular utility. Both trials showed that adding niacin to statin therapy provided no additional reduction in cardiovascular events. The medical and media response was swift. Niacin was declared ineffective, cardiologists moved away from prescribing it, and the supplement world used the failure to accelerate the pivot toward NR and NMEN. The problem with this narrative is that it fundamentally misreads what those trials actually tested, and the scientific community has been significantly slower to correct the record than it was to declare defeat. Both AMHI and HPS2, Thrive tested niacin added on top of aggressive statin therapy in patients whose LDL cholesterol was already well controlled by the statins. In this context, there's a straightforward ceiling effect. If cardiovascular risk is already substantially reduced by statin therapy, adding another intervention has limited room to show incremental benefit. The question these trials answered was not, does niacin work? It was, does niacin add benefit on top of already effective statin treatment in a population with already lowered LDL? Those are completely different questions. HPS2, Thrive, introduced an additional confound. The niacin formulation tested was combined with laropiprant, an anti-flushing agent. Laropiprant turned out to have its own adverse effects, gastrointestinal, musculoskeletal, and metabolic, that may have masked or offset niacin's benefits. Using a flawed formulation in a ceiling effect study design and concluding that niacin is ineffective reflects poor trial architecture, not a verdict on niacin monotherapy in appropriate patients. Niacin as primary lipid-modifying therapy in patients who cannot tolerate statins, or in patients with the specific phenotype of low HDL and high triglycerides that niacin addresses best, retains credible clinical rationale. The cardiologists who understand this distinction continue to use niacin in carefully selected patients. The broader medical community, shaped by simplified trial reporting and pharmaceutical industry interests, largely abandoned it. What this story teaches about evidence interpretation is essential. Does not work in this specific context is categorically different from does not work. Understanding the study design, the patient population, the comparator conditions, and the ceiling effects that may have constrained the results is what separates scientific literacy from headline-driven medical decision-making. Niacin didn't fail in AMHI and HPS2, Thrive. The trial designs failed to ask the right question about the right patients in the right context. That distinction is worth rehabilitating because it has real consequences for how we think about one of the most evidence-rich ND+ precursors available.